Hey everyone, I'm Crooks.
And I'm Ming.
Today we'll both be talking about the processes that help maintain homeostasis for plants and animals.
First is photosynthesis.
Inside the plant cells of a leaf, there are these little organelles called chloroplasts. Inside them is stroma and grana is the stack of thylakoids.
The thylakoid membrane is where light-dependent reactions occur. Take it away, Crooks.
The light-dependent reactions start with light energy.
Wait, but where does the light energy come from?
The sun.
And how is the energy absorbed?
The wavelengths of light are absorbed by pigments in the form of chlorophylls, the most common one being chlorophyll A. Another one is pheophytin, which is a darker pigment. But the actual start of photosystem is when photons, which is also known as light energy, strike photosystem 2. This causes the electrons inside of photosystem 2 to get incited, which means they gain energy.
What happens after that?
The electrons move down the electron transport chain, which is also referred to as the ETC. Then they go into plastiquinone and arrive at a cytochrome complex. And as the electrons move through the ETC, they release energy, which is used to pump hydrogen ions from the stroma into the thylakoid through the complex. Also, the transfer of protons with the electrons gained from photolysis creates a proton gradient that is used to make ATP from ATP synthase. Plastocyanin then takes the electrons and transports them into photosystem 1. Inside the photosystem is the reaction center complex. However, after all of this, photosystem 2 is low on electrons and needs energy. Photosystem 2 gains energy by splitting water, also known as photolysis. It splits the water, and the oxygen evolving complex is what actually causes photolysis. Photolysis happens to get energy. The water gives hydrogen ions, electrons, and oxygen, and the electrons go to photosystem 2, and then they get excited with more light energy. Once the electrons arrive in photosystem 1, they lose energy because they're giving it off while moving down the electron transport chain. Then they get the energy back through photons once again when light energy strikes photosystem 1. Now the electrons that are excited go down another electron transport chain until they get to the FeS complex. Using ferredoxin, the electrons transfer to an enzyme called NADP reductase. Here they are accepted by NADP+, then they combine with the hydrogen ion and turn into NADPH.
But isn't the goal of this to make energy? Doesn't ATP carry energy?
NADPH may not be ATP, but NADPH is also an energy-carrying molecule.
So what happened to the ATP?
Ming, do you remember I said something about a proton gradient that helps with ATP and stuff?
Yes.
Okay, good. As protons build up in the thylakoid space, they move back across the thylakoid membrane to the stroma through ATP synthase. This is called chemiosmosis. The energy that is released then turns ADP and a phosphate into ATP. This is what we call photophosphorylation. Now NADPH and ATP have been extracted through light-dependent reactions, and now they're going to be used in the Kelvin cycle or something. I don't really know anything about that though.
The Kelvin cycle? I know that. I learned about that a while ago. Okay, let me give you a little overview of what the Kelvin cycle does.
Okay, I can't wait to learn about the Kelvin cycle.
The Kelvin cycle makes sugar in the form of glucose from CO2 with NADPH and ATP.
Where does the Kelvin cycle take place?
The Kelvin cycle!
Oops, my bad.
It happens in the stroma, outside of the thylakoids. There are 3 steps: carbon fixation, reduction, and regeneration respectively. Let's start with carbon fixation.
Okay, so you said a CO2 is used. Is it just a CO2 or what?
No, there's a 5-carbon sugar called Ruby P, which is the acceptor that binds to CO2 in the stroma.
Okay, so is there like an enzyme that helps here, or does it just magically do it itself?
No, there's an enzyme called Rubisco. This creates a very unstable 6-carbon molecule.
Ming, what do you mean by unstable?
The bonds are very weak and they want to break.
Will do the bonds eventually break?
Yes, they break into 2 molecules of 3-PGA, and that's it.
Oh, okay. Is that it?
That's just the first step. The next step is reduction. The first thing that happens is that the 2 3-PGA molecules are converted into 2 G3P molecules. The energy used to convert these molecules comes from ATP and NADPH. One of the G3Ps leaves the Calvin cycle as food.
Okay, I understand it now.
The remaining 5 G3P molecules are reused in the Calvin cycle to make more RuBP and have more sugars leave the Calvin cycle. This next step is regeneration. Pretty much all that happens here is that the 2 G3Ps combine into 1 glucose.
Oh, nice. That was actually pretty easy. But, can you give numbers or something? Like, how much ATP and NADPH is needed to make 1 glucose? What are the exact numbers?
The reactants are CO2, ATP, and NADPH. And the products are G3P, ADP, and NADP+. A total of 18 ATP and 12 NADPH is needed to make 1 glucose molecule. Glycolysis happens in the cytoplasm. During the process, glucose is broken down into pyruvate and energy. Pyruvate is later used in pyruvate oxidation. The reactants are glucose and the products are pyruvate and energy. Pyruvate oxidation occurs in the mitochondria.
In the process of pyruvate oxidation, pyruvate, a 3-carbon molecule, is converted into acetyl-CoA, a 2-carbon molecule. Throughout this process, coenzyme A enters the process, NAD+ is reduced to NADH, and a molecule of CO2 leaves the process, all of these happening respectively. The products of pyruvate oxidation are CO2 electrons and, of course, acetyl-CoA that will be later used in the Krebs cycle.
As acetyl-CoA enters the Krebs cycle and combines with the previous oxaloacetate to create citrate, soon after, coenzyme A leaves the cycle. This creates a 6-carbon compound which loses a CO2, turning into a 5-carbon compound and reducing NAD+ to NADH. The 5-carbon compound continues through the Krebs cycle, losing CO2 and in the process reducing an NAD+ again, and also creating ATP from ADP and P, reducing NAD+ one more time while also reducing FAD, and then turning into a 4-carbon compound oxaloacetate.
Wow, that was a lot of information.
After that, the 4-carbon compound oxaloacetate receives acetyl-CoA from pyruvate oxidation and returns into the cycle again.
Oxidative phosphorylation. This one is a big one. Oxidative phosphorylation is basically where ADP and a phosphate become ATP. The first enzyme of it is NADH reductase. NADH reductase oxidizes NADH to NAD+, which creates the energy that powers a hydrogen ion through the enzyme from the mitochondrial matrix to the intermembrane space, and then to the outer mitochondrial membrane.
Wait, I know that mitochondria is the powerhouse of the cell, but is there anything that helps it create power?
Yes, ubiquinone is a coenzyme that helps mitochondria make power. Now, as I was saying, the second transport protein, succinate dehydrogenase, oxidizes FADH2 to FAD, which also powers another hydrogen ion through. The third transport protein, cytochrome reductase, doesn't oxidize anything, but it uses the energy of the H2O parts moving through to power the transportation of the hydrogen ions. The next protein is the cytochrome complex, which the parts of H2O also travel through, and it doesn't power a hydrogen ion through it though, because it doesn't go through the membrane. The fourth protein is cytochrome oxidase, which turns the 2 hydrogen ions and half an oxygen atom into H2O with the contents that came from cytochrome C, while also pumping a hydrogen ion through. And the last one isn't a protein, but an enzyme. ATP synthase pumps all the hydrogen ions anti-parallel to their original respective direction, powering the synthesis of ADP and a phosphate to create adenosine triphosphate.